And it might be simple or it might not be, but yeah, I think a lot of people are new to it, so yeah. So a biosensor, let's just go over the definition. It's a device that detects and reports the presence or quantity of a biological entity. So detection implies that recognition. So we're going to recognize typically through a biological or biochemical component. That's why you would immobilize something like an enzyme or an antibody or an aptamer or DNA that would selectively bind to metabolite like glucose or a protein of interest that might be linked to cancer, for example. And that's the recognition element. It doesn't have to be made by nature. It could be synthetic, like a molecular imprinted polymers and synthetic biorecognition agent. And then after that recognition, there's some sort of transduction into a measurable signal. That could be an optical or electrochemical transduction process. And the biological target, what's also puts the bio and biosensors, there's some biological target, usually some molecule, cells or viruses, bacteria, something that you want to detect or monitor or quantify in some way, shape, or form. And if you were to look at the journal Biosensors and Bioelectronics, it's one of the, maybe one of the more premier journals in the field. They define biosensors as analytical devices incorporating at least one of the following. So a biological material, tissue, organism, cell, receptor, enzyme, antibody, and so forth. Or a biological derived material, such as recombinant antibodies, engineered proteins, aptamers, which are synthetic DNA, or some sort of biomimetic material, something that acts like a biological component. So synthetic receptors, biomimetic catalysts,